Thanks so much for the opportunity to talk to you guys. I am a bit different. So I really don't like the idea of separate disciplines. When I see silos, I think of new opportunities. I see new domains to explore and new things to think about. So that's just one little start. So I come from, at my base, I'm probably an evolutionary biologist, though I've gotten to where I am via biophysics and neuroscience. So there you might see some echoes in there. And through that process, one thing that I've chatted a bit about this week is that I think physics and math and evolution, they all are trying to get at some elusive answers of truth that have commonalities. And so I think I'm really happy to talk to you guys a little bit about the evolutionary perspective and how we can gain some information from thinking about it from this perspective. And the last thing to mention going in, multi-scale collective intelligences, we are going to talk about many scales today. So you wanted multi-scale, I give you multi-scale, okay? Okay. So sociality in biology and life comes in many different forms and in many different scales. But generally you have loosely organized and uncommitted interactions in some situations, and then in other situations, highly interdependent cooperators who are obligately connected. And I'm really excited about how this happens. How does evolution build not just cooperation, but commitment? Not just sharing, but some sort of obligate interreliance. And so for those reasons, I'm really excited about these behaviors. So these are called social transfers. They're really interesting, juicy behaviors through which organisms transmit socially transferred materials. So this is like things like lactation or sex or other forms of parental care, laying eggs, all sorts of things. And so these are behaviors where something is made in one body that gets transmitted to another body where it acts. Okay? And these are ways for organisms to impact one another's fitness as well. And I think that's really the exciting thing here from a bit of a network perspective. If you think about what's going on within an organism, as we saw the slide with the tomato and all of the different molecules within it. So this is like those big clouds of molecules and things coming together, and things from one network get to impact the other network. And I think that's really cool. So these sort of behaviors, they have interesting ways that they vary. So for example, things that are transmitted are up here. This is the name of some of these funky social transfers. And you have things like partner relatedness in any given interaction. So there are some things where it's between, and I talk about relatedness because this is a really key variable in evolutionary biology, because it's sort of the basis of cooperation. You care about those who share your genes. And so if you're highly related to somebody, because we want the genes to get on to the next generation for fitness, you will do things that will benefit those who have shared genes with you on average. So some of these sort of behaviors are between like loosely cooperating individuals, and other ones where there should be a high reason to cooperate because they have high relatedness. And you can see other sort of things that go along with that. So some of these are separated in time. So mom lays an egg at some point. The offspring uses the resources in that egg some later time. So that's asynchronous. While in some cases it's much more synchronous exchange. So this one regurgitates, this one drinks. And they also can be brief or one-time events versus frequent. Now, these kinds of social transfers have been really impactful over the evolution of life. So I'm going to just mention briefly from our mammalian-centric perspective, our evolutionary history. So we split from like sort of reptilian things quite a long time ago. And then it's been sort of a relay race of different social transfers from eggs to lactation and then to the placenta. And basically now every mammal that exists, all of us, every mammal relies on milk at some point in our biology. And so this social transfer was the key to some sort of success that we've had, and here we are ruining the planet. So there are also other kinds of social transfer related success. So think about calcified eggs allowing terrestriality. So it's an important innovation. And the reason I think it's so interesting and so important is because it allows metabolic division of labor. What do I mean by that? So I think one of the very first talks had a nice sort of layout like this. Yes, yeah. This is the partitioning of a metabolic process into elementary tasks that are performed by separate units within a cooperative entity. Okay, so to think about this, there are actually quite a lot of metabolic tasks that are needed to run an organism, that many. So this is a metabolic network of how do you make biomass in something like us. And so each of these is a change from one thing into another thing. They're all different conversions to create different kinds of molecules and to transform different kinds of molecules.
Are these all, is it chemical reactions?
Yes.
These are chemical reactions?
There might be transporters in here as well, but for the most part, I think this is all chemical reactions. I think this is all chemical reactions.
And how do people put together this information? Like, how many years and years of work is this?
A lot, yeah. I mean, but it's incredible what we have now, and that you can take a sequence, and now we don't know this for all genes, but we know it for a lot of genes. And so you can say, okay, this enzyme, where does it fit? It fits right here. And so like a solid biochemist will be able to do that for you. And you can even figure it out yourself online. Just look it up. And there's like a little code for each of the enzymes, et cetera. So that's so much work. Because basically to do this, you need to make every single one of those tiny machines to do each of those damn transfers. And then you have to power them. You have to have energy to power all of those things. And so this is a simplified version. Now, it's a lot simplified. And all the steps require energy, right? You need ATP or concentration gradients, things like this. But now imagine that instead of having to do all of that yourself, somebody else did some of the steps and gave you an intermediate. And then you only had to do half the steps. You only had to produce half of the things. So this connection of these two networks like this allows them to specialize. Great. This division of labor is really key. And it turns out this happens very often in situations like parental care, where mom is providing all of these metabolic shortcuts to her offspring, so it doesn't have to do all that complicated stuff before. And so over evolution, offspring have become dependent on these processes. And so as you specialize, it builds this interreliance and this obligateness. And so part of why these care-related social transfers are so cool is because they allow this decoupling of resource acquisition and use over time, over space, across individuals, all through this type of metabolic division of labor. Yeah.
Do you consider also cross-species collaborations like that? For instance, are bacteria in our guts and...
I will come to them, yes. There is definitely metabolic division of labor with other organisms. Yes. So in thinking about how you evolve metabolic dependence, interdependence with another, so this is a story, a collaboration that I've been doing with Becky Kilner's group, and they work on these really gross little beetles that are called burying beetles, where they find like a carcass of a mouse in the woods, and then the parents, by parental care, so a mom and a dad, they turn this little carcass into a ball that they preserve, and so that's the first step of parental care. And then they lay their eggs, and then afterwards, the larvae hatch, and then they will feed them mouth to mouth through a social transfer. And then eventually everybody goes off their separate ways. So what Becky's group did was to evolve these populations either with and without the social transfer care for like 50 generations. And then we split them and reared each in different care environments. And what we found was basically here that when we had this population that was reared expecting care, was reared without it, they did not do very well. They generally died. And then when we looked at actually what was in their guts, we could see that they were actually really poor. These ones full to care to no care were very poor at actually assimilating any of the food from the mouse. And so it seems, And so it seems that the offspring have evolved this dependence on something that the parents are providing, some sort of shortcut, metabolic shortcut, and they evolved that dependence over just 50 generations. So this is this entrenchment of this interdependence. So that leads me to the question I think is even more juicy and multi-scale, which is when does this metabolic division of labor lead to what are called major evolutionary transitions in individuality? So these have been at various times termed phase transitions in biology, but basically it's when you go through this transformative step, shifting from just a bunch of folks who are cooperating, a society of cooperating individuals, into a new level of individual. So a new self. So here's the borders of the self that contains these individual units.
And we would put some kind of parenting relationship between solitary and cooperative?
Parenting?
Yeah.
No. Well, I mean, parenting, okay. Parenting doesn't map on this so well.
Okay.
So you can have...
Even the facilitation of eating by the parent, you wouldn't count it as some kind of social?
You could have parental care doing individuals in this category, or this category. So yeah, they don't have to be. Yes. Okay. So what do I mean by major evolutionary transitions? Here's some examples. Here's some of the classic examples. So genes coming together to form genomes that reproduce in a packet, simple cells engulfing mitochondria or chloroplasts to become more complex cells, single-celled organisms that happen to stick together enough to become somehow a new level of organism, and then my favorite of a bunch of multicellular organisms that hang out together enough and somehow manage to become a superorganism. Now, these can be categorized into two different types of major evolutionary transitions. So there's ones where it's the equivalent pieces that come together and stick together. And then there's different types. And so this is like between different species. And actually, so the major evolutionary transitions that are in the sort of like type fraternal category are relatively few. And there are hundreds of cases of the unlike type. So for example, many different insects have endosymbionts that live inside them and allow them to access new niches, new opportunities. Yeah.
So is the idea that you have a new self and they reproduce or die together? Is that the
Yes.
time at which you're going to have a new self?
Their success is linked. Yes. So like if a cell divides and it has a bunch of mitochondria inside it, there's those mitochondria, they're linked. Yeah. Yes. And so metabolic division of labor, how does it interact with this major evolutionary transition sort of way of thinking? So there are examples of metabolic division of labor that are just facultative. And so actually a lot of microbes, a lot of bacteria, one type of bacteria has waste products that are delicious and important for somebody else, and vice versa. And so you have these cross-feeding interactions. However, they're uncommitted. It doesn't matter who's going to make that molecule for me as long as I get it. Yeah? And so oftentimes you can sort of swap out as far as I know, every time you can swap out the members of these microbial communities. But there's a lot of really cool theory and experiments on microbes in that space. And then within the sort of like-type evolutionary transitions, there's all sorts of cool types of metabolic division of labor that happen within your body. So, I mean, basically, people who study physiology can tell you a lot more about that. But for example, your neurons are really costly, and the rest of your body supports them. Same with your germline. Cancer exploits that. And then in these sort of unlike type, as I said, you have this whole category of the endosymbionts, but you also have mutualisms that can become quite important obligate forms of metabolic division of labor. So I think these are fascinating, and it leads me to the question of whether there are sort of common principles of how these things arrive, how are they governed, how can we understand them. And so I've come up with a few sort of principles that I think tentatively might be important for the evolution of collectively developing systems. One is that they need a networked private channel for resources and communication between the different units that enables this metabolic division of labor. So it needs to be a private channel that it's not shared with non-cooperators, and it needs to mediate this metabolic division of labor, and it can't just be between mom and child, for example. That needs some sort of networking across the many units of the organism. It is the interdependence that arises through metabolic division of labor that sustains this cooperation. And this one gets a little bit more in towards the evolutionary biology, but it's basically shared goals and shared priorities. So reproductive conflict, in the case of evolutionary biology, needs to be held below a given threshold to really initiate such a system. And so I'm going to go through a handful of things today within the time and try to point out where we connect to any of these ideas. Okay, so one thing to sort of start out, to tell you a little bit more about the biology of the systems I'm going to be talking about. So I think it's really important to think about what are the edges of the self. What is the cooperating unit? Where are its limits? And for a multicellular organism, it's pretty clear. I mean, you see the edges. And you have this separation between germline and soma. So ovaries or testes, that's the actual part of you, of your cooperative unit. So I'm made of a whole bunch of cells, but I'm still one me. And the only thing that's going to ever potentially reproduce, or has reproduced, I have one child, is my ovaries, that's it. And the soma, thankfully, is not going to reproduce. With a superorganism, you have the same sort of story, where the queen is acting as that germline. She also has a male who she mated with, whose sperm she sort of keeps in her pocket and continues using over time. But so you can think of them together as the germline. And then everybody else in the colony is just like the somatic cells that make up your gut or your skin. And so in these sort of situations, we have high cooperation, low conflict. Usually the different units are maybe clonal or at least very highly related in the case of ants. And you should have strict reproductive division of labor. So the only thing that can reproduce is the germline. However, there are a lot of weird cases of ants where that breaks down, and that is not the case. So, for example, most ants are like this, these gigantic queens and tiny little workers. But then there's a lot of other ants out there where you have queens and workers that are indistinguishable. The reproductive is morphologically just the same, and you can have workers that can become queens. There are a lot of other different sort of biologies that can function here, in which case you don't have this sense of a collective that has shared goals, because everybody has the capacity to enact their own will or priorities. And I mean, I'm telling you guys all of this because these ideas about how do you, organizational rules, how do you make collectives, how do you enforce collectives, really? And so these are things about how that's worked in biology. Okay, so social insects are super cool. They have many of these different social transfers that can allow metabolic division of labor, but there's only really one that allows this sort of, yes, networked transfer.
I was just wondering about this. You said you need this sort of strict division of labour when it comes to reproduction. Why is that necessary if they're already clonal? Like, why would it be such a problem if different parts of your body could reproduce? Because we've still got the same interest. If my hand can reproduce, as long as it's still the same DNA that's going to be reproducing, it wouldn't necessarily be a genetic conflict.
So it's control of cheaters, I think, because why have microbes? Microbes are, in a community, they're pretty much clonal. Why have they not become multicellular organisms? Why did they not go through this transformation? And I think the answer really comes in this autonomy and in constraining autonomy. And so when every unit's reproduction is linked to the whole, shares in a company, for example, that's, the company's success is your success. Like that enforces that. If that's the only way you can succeed, that enforces the cooperation. Do you see what I mean? So if it's clonal, it is clonal to an extent. But if just one mutation comes out and, changes something. So for example, in like the clonal raider ants, which you may be familiar with, there are some ants like this, and you can have cheater lineages that do arise and that can bias themselves towards reproductive behaviors. And then you're just very susceptible to cheating. So I think that's why it's important. Okay, so trophallaxis is this beautiful behavior. One ant regurgitates, the other one drinks it. Great. And some ants do this, a lot of them don't. Well, most of them do, some of them don't. And as again, this is in major contrast to other social transfers like milk. And so it's neat because we can see why some do, why some don't. And so this behavior is really beautiful, fun. So this is a colony of honeypot ants. If I can figure this out, there we go. And I'm just going to speed it up because it lasts for a while. So they have some, first, they have some food sources here, and I think down here. This one's more delicious up at the top. Over time, more and more of them get this tasty food, and we're seeing through their abdomen and seeing them passing around this fluorescent stuff. And so over time, it gets rapidly disseminated, and we can watch how it goes. And so it creates this real network over the colony, and the colony has its own social structures, and so there's interesting spaces to explore transmission over social structures there. But that's not what I'm going to focus on today. So this fluid can transmit a lot more than food, though. It transmits not just food, but all of those molecules, those giant pathways. It transmits physiology. You guys don't care about exactly what's in there, and that's fine. And I won't tell you. But one cool thing about it is that from all of the little bits in there, we can figure out who the ant was and other conditions about it. So we can say, okay, it was from a mature colony. It was a nurse. It was from a field population. And so we can see all of this kinds of variation based on the set of tiny little machines that are in its guts. And so it creates, yes?
I have a question. So these ants are technically siblings?
Mm-hmm.
And do they pair up selectively, or do they do this with multiple different?
As far as we know, they do it with everybody. And there's a little piece that I'm going to tell you about that later. So actually, this is my introduction about what we're going to talk about. So I'm going to tell you briefly about this is the longest part, what makes a social transfer evolve. Talk briefly about transmission networks and so different forms of social organization, so the ant governance that we talked about. I'll talk a little bit about endosymbiosis and metabolic independence, adaptive blindness, and that's when I'll talk about who they do it with, and then a little model at the end. So, so some of the species do this, and some of them don't do this. Why? And what can we understand about why one of these behaviors would evolve in the first place from that? And so let me explain to you what this is. So this is a phylogenetic tree. And so a phylogenetic tree is like a directed acyclic graph. This is the ancestral state, the root of it. And each of the early branches are early separations from that. And then each of these branches is sort of a speciation event with two different paths, and then further diversification. And around the edges, you have the extant species, so the species that are still alive. And in this case, this is not actually species, it's a genus level tree, but whatever. The point, so here I'm talking about, this is different groups of ants, and then these are about a behavioral trait, is whether they do this behavior or not, and in how many species have they been annotated to do this.
There's a red color and a purple color.
So we have, yes, so that's true. This is more subtleties, so you don't really need to know. But this was just the first analysis of what we could find in the literature about this. And so you can see on average, the ones in this big group, so there's a big separation in the history of ants between the poneroids and the formicoids. And mostly one group does it, but there's a bit of yes and a bit of no. Why? So there's a bunch of traits that loosely correlate with this behavior, and we're trying to figure out why this happens. And so, yes.
So, Adria, when that picture, so is it actually like experimentalists have studied every single species over there and determined whether it does trophallaxis or not?
Yeah.
Is that how it works?
Yeah. So in each of these, so for example, here in Ecophylla, that's a weaver ant that you may have seen in Australia. And there are 2 species within Ecophylla who have been shown to do trophallaxis. And so, yeah.
So how does one find all of this data? It's just like scattered out there in the literature. You read every single paper? Or
You comb the literature either with technology or with your eyes. Yeah. Yeah, and so here, this was just based on the literature, this first hunt, to find out what was there. And then you can do the same thing, essentially, for a bunch of other traits that have been associated with this. So just the logic behind them is that ants that drink some fluid, they're usually the ones who do trophallaxis because you don't have a bottle to bring fluids around, so you have to do it some other way. Usually ants that have a sting don't do it. Big colonies tend to do this behavior, and certain phylogenetic groups don't. And so we built a bunch of different hypotheses over why this might be happening. And then we collected all of these traits for all of those species and more to figure out which of them is more supported over the evolutionary history of the ants. So the transitions from one state to the next across the phylogeny. And so basically, just to walk you through some of the ideas. So the first is this eco-opportunism idea. So basically that as the earth changed and became more rich with plants, with angiosperms, there were more sugary liquids. So there was this opportunity to be seized, and ants needed a way to move things around. So they evolved trophallaxis, and then the sting, they wanted to expand their gasters like this, so they got rid of the sting, and then it was so successful, so they got larger colonies. This is represented here in the little graph. Yeah. The next idea is this reproductive conflict hypothesis. So this is about the importance of shared priorities. And so in the average ant, the queen can do everything. She can make workers, she can make males, she can make queens. Workers can usually either only make males, who are essentially seasonal, or they can make nothing. But as I mentioned, there are some ants that can actually do everything in some species. And only certain species have this sort of autonomy that allows them to sort of cheat the system or bet hedge, depending on how you think about it. And the issue here is that why would you cooperate if you could have your own babies? In some cases it is advantageous to cooperate. In many cases it's advantageous to cooperate with people who are not your offspring and with whom you're not going to have babies. But still, if you could do it yourself, you know exactly whose babies they are. And so this, to me, is really a separation between not superorganismal ants and superorganismal ants. So shared priorities and non-shared priorities when they have this super worker who can do everything. So there's more conflict within the societies. And so in this hypothesis, the idea is that, and this is debatable. This is not really clear from the literature, this point, but it could go either way. The common ancestor of all ants could have had totipotent workers, so workers who could do everything. We know they had true queens as well, but basically they could have had cheating-capable workers. And then once totipotent workers were lost, as the lineages evolved, then workers were maybe more willing to share resources with others because they knew they were in cooperation. And then this led to trophallaxis, which led to larger colonies. And then once trophallaxis was possible, they could do sugary liquids and lost a sting. Then lastly, the last two hypotheses are a little shorter. So social complexity, basically larger colonies need more communications between the individuals. And so perhaps they needed this trophallaxis as a means to share information. And then, but colonies became large for other reasons. And then the other was that there was maybe morphological constraints. And so basically we collected all these traits and tested them on the phylogeny using a phylogenetic path analysis, which is essentially like normal path analysis, but constrained by phylogeny and the shared evolutionary history.
Are there any instances of kind of convergent rediscovery of some of these clusters of traits, or it's strictly phylogenetic?
Yeah, there are. Yeah, I'll show you that briefly. So essentially, when we did this, these two hypotheses were both better, pretty good predictors, and so we merged them, we averaged them, and this is sort of the average showing the effects and how strong they are. And so, for example, we see that the presence of these super reproductive workers strongly inhibits the evolution of trophallaxis. Sugary liquid diets and trophallaxis promote each other. We can't tell what happened first because they happen at the same time. And then large colonies arose after trophallaxis. So that's neat. But and also we found using something called speciation and extinction analysis, which looks at where there are branchings and where there are deaths and extinctions, that having this behavior, this sharing behavior, so the more sharing in the colonies increased net diversification, so more splits and more losses. So to figure out what was the ancestral state, what was the history of this, we needed a lot more data. And so to do that, we imputed data with machine learning. So basically we had all of our traits, we built a model, Bayesian neural network, and then we fed it all of the traits but trophallaxis and had it predict trophallaxis. And it has its various uncertainties. That's the value of doing it Bayesian. And we checked these with actual experts who studied these species but hadn't published about them, and it was decent accuracy. And so then we can reconstruct these ancestral behaviors and see how they change over time. And so I'm not going to spend too long on the ancestral state reconstruction, but just this is, we use something called SMAP, which is a stochastic mapping approach in a Bayesian framework, so it can propagate the uncertainty into the tree. And so whether they had sugary liquids in their diet, so again, just to orient you on the tree, so the probability of this trait is in color, and the root node is there, and so we're pretty sure that it probably didn't do this behavior. It probably didn't eat sugary liquids, the ancestral ant, 150 million years ago. But then over time, we can see when it got gained about here. And there were some losses and things like that. And then for worker reproductive autonomy, so whether they could reproduce themselves, we can do the same thing. And this was a lot trickier, and it was super sensitive to the priors. So the first time we did this, we used priors that were constrained by the true transition rates, and the transition rates strongly indicate you only lose reproductive capabilities. So cooperation gets entrenched. And so we got a very certainty that the ancestral state had fully reproductive workers that way. Here we've loosened the priors, and so it's more conservative, and it's not totally sure, but still the transition rates are very, very clear that you, for the most part, only lose the capacity to reproduce yourself. And then for trophallaxis itself, basically when both conditions were met, where they had sugary liquids in the diet and they didn't have reproductive workers, that's when they would evolve this behavior. And so essentially what we can think about that as is that once you have a reason to connect bodies and you're willing to do it, that's when you will evolve something that actually merges the bodies in such a social transfer. So thinking about organizations, not sure exactly what the parallel would be, but essentially you have to have the motivation and the desire. You need to have enough cooperative desire to work with the other and a reason to do it. So then I'm going to briefly talk to you about a few other little stories. Okay. So this is about metabolic division of labor. And there are a number of different ways that different ants do it. So I told you about some of these different ways. There are some really creepy sets of social transfers out there. And so we think that the types of metabolic division of labor and the interreliance across them are going to vary. And so we're building these genome-scale metabolic models of those giant biochemical pathways. I'm not going to tell you too much about it, but I'm going to show you this creepy behavior. So these are a very weird ant where you have an adult that feeds on, so the larvae end up having a tubercle on their ventral side where they secrete something to the adult, and the adult drinks from the young. So it's like the opposite of normal parental care. Are we not starting the video? There we go. And so you can see her doing this right here, okay? And this is, yeah, so these larvae and the adults, they eat raw prey, they don't do trophallaxis, they don't have true queens. They're actually, it turns out, Hugo has dissected a lot of reproductive organs and figured out that they are not even, they're clonal. And they only lay one, they only have one egg layer at a time. So they're figuring out their governance structure is weird. So thinking about, so because everybody's clonal, everybody has the capacity to reproduce. So what actually controls who is the reproducer, who is the one who lays eggs? And so we put barcodes on the ants and we watch them and we see how they move. And one of the things we can see is that basically this egg layer is monopolizing access to this larva. And it is the material that the larvae are passing to the queen or to the reproductive that allows her to lay eggs. And so if we give a colony that has no brood in it one of these larvae, they will lay eggs. And so it's like, it's the secret to their fertility. And so it's a very different governance structure to a normal ant colony. There are all sorts of other fun ways to do this, and we're trying to track the transmission.
In humans, the closest thing is sortition, random assignment of leadership.
Cool. Yeah. I don't know if it's random. They do fight when it's time to decide who gets access. We're also trying to figure out what is actually the currency of this, what is in this stuff. And so to figure that out, we're collecting like all of the genes and proteins that are made by all these different parts of ants, and we can basically filter them to say what are the things that the larvae are producing, and then we filter from those, what is it that the adults are not producing of all of those genes, and then what is actually in the transmitted fluid, And then what is it that the adults actually have in their guts that they're drinking? And yes?
If you artificially feed this thing to two ants, do they both start laying eggs, or is there competition between?
If they're together, we haven't tried
Same
that.
colony, if you just feed two different ants, force feed them this stuff.
We haven't tried that. It's hard to force feed. Okay. And so then we figure out what is common across all those things. And what we find is that there are these storage proteins that larvae are producing. There's also some hormones. There's interesting things that we're trying to figure out from a molecular side. So I got my 10 minutes, so I'm going to move through the other pieces, which are interesting. So endosymbiosis. So this is an example of like two becoming one, two separate species. So if you think about little microbes, they're in your gut, they produce things, you eat, they're good for you. There are cases where it can be very facultative or just sort of convenient, but then there are cases where it becomes obligate, where this other thing it becomes engulfed and it becomes a requirement for everybody's reproduction. And can these actually go from a sort of lightly conflictual situation of a bit of tit for tat? Can those become a mutually, absolutely obligate relationship? And so you can have these unidirectional obligate situations where the microbe needs to be inside the host, but the host doesn't really need the microbe, and it's actually kind of inconvenient in often cases. And then there's some cases where it's bidirectional, where the host actually cannot, like the ants that we mostly work with, they require an endosymbiont, and they will die if they don't have them. And so thinking about these things, we can build these models over the whole genome and to say, what are they capable of, and what do they have in there? And so we have our red ones are the bidirectional, blue is unidirectional. And so we can say how big is the genome of these different types of things? And how many genes are in there? Okay, so it's very correlated. But then when we start looking at how many metabolic genes are in any of these, so these ones which are unidirectional, who are kind of like parasites, they have all of these other genes that aren't actually doing metabolism. So they do a lot of other things, but not something particularly useful. And you can do the same thing with reactions, the reactions they're capable of. And you can again see that even these tiny little genomes can do more reactions than these more parasitic guys. And then when we look at, they make all of the useful things, they're great. And when we can even look into what they're actually doing, what we can see is that the ones that are sort of cooperative, this bidirectional thing, they are just producing useful things, but not so these things that transport or these binding things, they're associated with stealing. And so basically we can get genomic signatures of what are the cooperative genes and what are the anti-cooperative genes, which are about taking from the host. So that's getting a bit towards this cooperation and conflict. The adaptive blindness. Okay, so this one I think is going to be more up your alley. So within a colony, what is it, Ida said? Is that what her name is? She was asking about what if they do this preferentially with siblings. And so if they did, what would happen? If they could preferentially associate, that would be pretty risky because it would allow sort of sub-pockets to form. You can have problems, and it would could potentially break down the collaboration. And so we really wanted to know if the ants could perceive what was in the fluid they're passing around. And so this is a master's student who basically we were using this little choice assay where there are two different foods, and we can measure when they interact with them. And so we get a nice signal, and we can look at how they make their choices. And we could measure all these different things. And so here's the two croissants that we saw earlier. We gave them two things that were exactly the same, and we can see their preference. Some of them chose one, some of them chose the other one, some of them tasted a bit of both. If we give them a clear choice, like delicious sugar versus no sugar, they make a clear choice. And if we give them a sort of artificial trophylactic fluid that we make that doesn't have a lot of interesting things versus one that has some protein, the same thing, but with some extra proteins, they make a clear choice. So they can see all these things, and we can watch it. But then we wanted to know, what about the things that are important to them in their decision-making processes that impact, for example, larval development, the good stuff. And so, for example, here, when we gave them this important hormone, they don't tell the difference. When we give them these small RNAs, they don't tell the difference. When we give them hydrocarbons, which are like the smell on the body that they can use to tell friend from foe, they can tell the difference. They see it. But then I think the biggest sort of challenge we gave them was to give them a choice of either their own little molecular machines versus something purified from like cow's milk. And to say, do you tell the difference between these two things at the same concentration? And they didn't. Neither foragers nor nurses, so different categories. And so to me, what this shows is that basically all of the important signals that could manipulate or modulate colony life, they don't see them. And so essentially any sort of voting they do for the colony's future in terms of how they rear their larvae or how they pass things around, they're blind to it. And so they're just following the social networks and not manipulating them, which I think is pretty neat. Last one, I'm not sure I'm going to get through much of this, but basically the idea is that how does metabolic cooperation evolve in a changing environment, as we saw. And so this is a model that I've been developing with Simon van Vliet in Basel, and it's about metabolic flux. And basically it's a death-birth model where we have individual ants that go out, they collect resources, the colony grows, and it reproduces, and it has fitness. And we can let their preferences evolve. And the major difference is that when they gather protein food, like predatory ants would do this, so the ancestral ant would just eat like a dead cricket. That requires no metabolic work. Well, if they want to adapt to start using sugar, which is actually a terrible resource and a terrible, terrible thing if you want to build an ant, then they have to do metabolic work to do that. And so can they get over that barrier to evolve that preference? And under what conditions will they do it? And we let the queen's contribution to the metabolic division of labor be this variable about whether they've evolved it or not. So can they subsidize the queen, basically? And so sort of somewhat as expected, the way we built it is that eating sugar is harder. And so indeed, eating sugar is much harder. They do very poorly, whether we use linear scaling or 2/3 scaling. But when they evolve metabolic division of labor, they can manage. So the model is doing approximately what it's supposed to do. And then when we add in ecological variation, it gets a bit more interesting. And so the orange dots represent what the ancestral state was, what they should have been eating and how they should have been behaving. And so what we see is basically when we have low resource availability up here and high resource availability, and then in the middle, we have this sort of biased availability. And so sugar specialists, this sort of hard thing, only really evolves when you have this sort of biased availability. Okay, but in order to do that, you need to evolve metabolic division of labor. Yeah, and then this was the more interesting part. So we built an evolutionary model of this and put different colonies in the model to allow them to succeed, what the different preferences succeed or fail. And we had all these different patterns with sometimes branching, sometimes specialization in one, sometimes it changes, sometimes it doesn't. And we allowed different kinds of trade-offs and different food availabilities and resources. And basically what we found was that when we only found branching when it was a very poor environment. And so they only would evolve these different strategies when they were really in deep competition, and they needed to evolve this sort of, they needed to innovate out of this hard place. And indeed, that might be what we find, and why you find these kinds of ants only in the tropics, blah, 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 blah. Okay, so just to close up, yes, so we talked about these principles for collectively developing systems. And so we talked a number of times about this private channel and when it would evolve, when it might need to come about, what you need to do it, seen ways that interdependence comes about, and also about reproductive conflict. And so I think there's other cool organisms that we can think about to explore this, for example, corals and tunicates and other neat things. Yes. Given that you guys all like games, not all of you, if the organizers are interested, I have run improvisation games for scientists for like 15 years. And I have some games that I think you guys might be interested in. They're not hard, but they're about collective behaviors and collective choices. And so if there's a time, maybe after the talk or maybe tomorrow, that the organizers would be interested, we can just do it just in the coffee area. So I just put that out there if you guys are interested. And so with that, I thank everybody who contributed to this work, my funders, lab members, collaborators. I'm happy to take any questions.